Hello, students, and welcome to Unit 2. So I've got some requests for Unit 2 videos. Some of my students really liked that I went over the unit guides for Unit 1 and some of the key ideas. So let's try to do a little bit on Unit 2. So before we get started, I would like to point out that this unit is weighted a little bit more than Unit 1. In fact, I noticed looking at some previous exams that the Unit 1 questions, it says 8 to 11%, but I found that that's for both multiple choice and FRQs. I think for FRQs, I don't see very much of Unit 1 incorporated. Seems like Unit 2 is more often in the FRQ. So this is definitely an important unit for your AP test. At the end of the unit, you'll have access to some practice questions as you study for your test. But let's get into it. So what should you know about Unit 2? So it says the cell is the basic unit of life. A lot of these things you already covered in biology, but it's a matter of refreshing it, reminding yourself what are the different organelles in cells. And it will be a focus on, there will be a focus on eukaryotic cells. We'll occasionally mention the analogous situations for prokaryotic cells, but keep in mind, as humans, we are going to have a bias towards eukaryotic cells, cells that have specialized organelles. And it's not that prokaryotic cells can't do things like photosynthesis. They have some of these abilities. They have ribosomes. They do have a lot of these same abilities, but they don't have these specialized organelles which allow for more efficiency. For example, when we think about ribosomes, it's incredibly advantageous for your cells to have ribosomes right next to the nucleus. We have this REF-ER that's bound to the nucleus or attached to the nucleus, and that allows for quick transportation from that mRNA to a ribosome. Pretty cool. They want to emphasize that the cell membranes control the exchange of material with the cell's external environment. Of course, this is the whole purpose of having a semi-permeable membrane. Different cells have slightly different types of membranes. For example, in plant cells, the cell wall is a cellulose-rich extra layer in the membrane, whereas animal cells have the phospholipid bilayer. So it looks different depending on what the cell is, but all cells have a semi-permeable membrane. They need to keep certain things in, like their genetic material, and keep certain things out, like a virus's genetic material. This ability or lack of ability in terms of it getting infected by a virus or being affected by another pathogen, the breaking down of homeostasis, this is a central idea in biology. The maintenance of the conditions is called homeostasis. Of course, there's many definitions for homeostasis in this class, but this is what the one we'll think about. If we're looking at a cellular level, so for this unit, our definition is the maintenance of the right conditions to keep a cell healthy. And this is laying the groundwork. We don't cover everything about cells here. In fact, you could say that our whole semester is pretty much about cellular biology, because next unit we're going to talk about cellular energies, and then we're going to talk about cell communication and reproduction and replication, sorry. Not reproduction. Reproduction is for animals and larger organisms. Replication in the cell cycle is for cells. But everything we cover in this unit, all about cells, up until almost the end of the semester. Okay, let's see what they suggest. This is similar to other things. I do suggest you print out the science practices that you need to know for the test, and also what sort of prompts are asked. but I will give those printouts to students I have in class soon. Okay, on the exam, students frequently can correctly identify organelle, but fail to describe its function. Yeah, of course, if you just memorize what are the organelles, that's no good. That's not gonna help you at all on the test. So we're not gonna say, what is this organelle? We'll move beyond that sort of middle school model of just memorize the parts of an organelle. Now we need to actually use the vocabulary, use the names to really understand not only the function, like the mitochondria is the powerhouse of the cell, but what does that actually mean? What's the actual chemistry involved? What are the actual roles? And how does specific structures in the mitochondria allow for its functions? For example, in the mitochondria, let's pull that up. We have the cristae. Mitochondria. One second, I gotta blow my nose. Probably when I find this picture, I'm gonna cut the video to introduce our first unit. But if you're watching the whole video, you got to learn a little bit more about the unit guide in general. Let's just finish reading this before we focus on section 1. So last things that you are supposed to know for the test, I'm supposed to teach you is that students should be able to explain the relationship between structure and function on both a subcellular and cellular level. What does that mean? Subcellular means smaller than a cell. And so the structures of organelles, but also the structures of a cell as a whole. So a big idea such as the central dogma of so-called central dogma of biology, the DNA is used to make RNA, and RNA is used to make proteins. That's a cellular level. That's happening in many different parts of the cell. Cell is coordinating that as a whole. Whereas subcellular could be something such as photosynthesis that happens in a localized area of the cell. Avoid using catchy analogies and food-based models. Okay. I guess fortunately I don't know these catchy analogies, so I can't misteach you, but AP wants me to know, don't use food-based models and say organelles are like foods. I've never even heard of that, so that's good. I'm not going to teach you that. The graphing skills learned in this unit are important. Yeah, we've already started that in class, but be aware. Now let's pull up that FRQ one more time for a reminder. This last year, and this happens for most AP tests, we had a prompt where you were required on the AP test to graph with error bars. Let's find that one. Is that number 1? Graph the data with error bars. Yeah, number 2. So on the 2012, the AP test that just happened, students were required to graph with error bars. And what the chief grader said in their video is most students got these points. These were some of the points that most students got. However, some students forgot to do the error bars and lost a point. So make sure you understand how to graph error bars. And some students forgot to label their axes and lost with units and lost a point. So these should be automatic points. If you're asked to graph this, we're going to do it so many times in class. And if you're not doing it enough in class, just grab an AP book, get some extra practice graphing. There's some really good AP books like 5 Steps to a 5 that have extra practice. But depending on your background in math, this might be very easy for you and you might not need any extra practice, or you might need some extra practice. So make sure you're able to do this. It should be the easiest points you get on the AP test. Just graph. That's all you have to do. So what they're saying here is that graphing is essential to this. Students often fail to earn points because they draw error bars incorrectly and fails to use them to draw conclusions. So graph with units and understand what that means. Okay, now to section 1. So let's look at the mitochondria together. Okay, so welcome students to section 1. So we're going to talk about the mitochondria in our discussion of structure and function. So that's a pretty cool picture. Let's look at that one. So as we're going over the different organelles, we're going to talk about the most important ones. First the mitochondria, then the chloroplast, then I will just make a video on a bunch of different other ones. So first the mitochondria. So what do you need to know about the mitochondria? Well, as seen here, this doesn't look like a normal organelle. It has the typical membrane, just like the cell itself has a membrane. Organelles are membrane-bound parts of the cell, and so we have this cell membrane, but inside of it we have another membrane. So this is a very unique structure that's double membrane. This is not the case in most organelles, but here we have a double membrane. And so we have a double advantage here. Here, because of the geometry and this membrane, we have incredible amount of surface area. Why is that good? Well, in each part of this inner membrane, we're able to generate ATP. So the more curved it is, the more surface area, the more effective it's going to be at this. This is, these curves are called cristae, which is Greek for something, and the inner fluid is called the matrix, and this is really where the ATP synthesis is generated. Don't try to just say this is the powerhouse of the cell. We're moving beyond that now. We're actually understanding, what does that mean? Well, that means in the mitochondria, in the membrane, we're able to use the surface area of it and its unique structure in order to generate ATP. How does it generate ATP? Is it just made out of thin air? No, of course not. Usually, especially in humans, what's happening is we're breaking down glucose. We're breaking down our macromolecules and using those macromolecules in order to make the ATP we need. Just like, and we'll talk about that generation. Where does that glucose come from ultimately next time when we talk about the chloroplast. But I just want to briefly introduce to you that the theme of this whole unit is understanding the different structures of the cell, whether they're subcellular, like an organelle, like mitochondria, or whether they're a process in the whole cell, such as transcription and translation, that involves many different parts of the cell working, coordinating together. So ATP synthesis is something that's specified, it's localized in the mitochondria, and it's enabled by the unique structures of the mitochondria, which allow for a lot of surface area. Of course, other cells, let's compare this to a prokaryotic cell. Prokaryotic cells also need ATP, but they're much less efficient at it because they don't have this specialized ATP synthesis organelle. ATP synthesis is going to have to happen by breaking down glucose just in the cytoplasm of a prokaryotic cell. Definitely less efficient. Okay, that's it for today. Have a good day, everyone, and I'll see you next time.